Log on, tune in, find out. Another good idea from Cambridge.
Study. So that's a specific phase of drug development, and we hope that we put a schedule and an agenda, a very nice program, people from both academia and industry. We expect that we will start with some state-of-the-art presentations on the advancement in the methodology, and then we will call here at the podium the representative from the industry explaining what are the challenges in the practice with implementation and maybe even developing and solving some open problems in design of experiment coming from the practice. And then we will end up in the late afternoon with 3 case studies, real case studies from pharmaceutical industry, where people will present, or presenters will show you the attempts and solutions for these challenging problems. And then at the end, we have a kind of a panel discussion. Will be very free, entertaining here. All presenters will be asked to participate in that one, and it will be monitored by Mike Cramps. So I hope that we will have an enjoyable day today. And let's start with the first presentation. So I'm presenting myself. That's the topic of my presentation. Adaptive dose ranging design with 2 efficacy endpoints. So let me see if I can. Yes, works. So this is a brief outline of my presentation. I will start with the motivational study for this type of investigation or research. It's called cognition impairment associated with schizophrenia. It's a specific type of disease, and it's like something which now it's very new as a disease, and there are different attempts to develop drugs for this type of indication, and there are some challenges, and I will just briefly describe them to you, and then I will suggest a solution to that kind of challenging design problems in this indication, and we will be talking about adaptive design. I will describe you adaptive design which we considered in order to address this type of problem. And then I have some slides and on the operating characteristics of this adaptive design. This is how you usually propose a design. It's very good to have an analytic solution, but unfortunately you will hear today once more that the practical problems are very complex, and unfortunately we don't have those analytic solutions so far. And then the only way of looking at the property of the design is to do simulations. And then in order to convince the clinical team, in this case, that this design has some rights of being considered, you have to compare with something. In the first session, I think yesterday, but more precisely a week before, it was another conference here. And I think that Val Federer was saying that if we have a theoretical solution, even if we will not apply the theoretical solution in practice, it's good to have it benchmarked. I think that here, in my case, I don't have that theoretical solution, so I will use a benchmark, but that benchmark will be a traditional, let's say, approach to this type of problem. And we'll try to see what are the pros and cons of one versus another. And then I'll finish with conclusion. So, what is this cognitive impairment associated with schizophrenia? This is like I said, a new type of disease, and the challenges in drug development for this indication are the following: that a successful therapy in this indication is supposed to be a promise for overall patient being, but that overall patient being should be measured not in just one endpoint, one co-primary endpoint, but with 2 co-primary endpoints. So it's not only symptoms that the patient will be kind of feeling that they feel okay, but also some cognitive, improvements in their behavior. Subjects are clinically stable in the there's a category of schizophrenia score on a previous antipsychotic, and the study will be administered as an adjunctive therapy to this antipsychotic. So it's not placebo versus active control, it's standard of care and then I'll tell you, you would like to say that your drug on top of standard of care provides some improvement. So no formal regulatory guideline exists so far. However, there are a lot of cooperation between academia, some governmental organization, FDA, at least in US, and I have a reference here to the recent FDA NIH matrix guideline for clinical trial design of this type of indication. And you can see here that they are putting like what do we know in the last 5 years. So it's indeed a very new type of indication. The phase 3 study requirements now will be to have a 6-month treatment duration and show a clinically meaningful effect of standardized 0.5 standard deviation. So both in cognition and this functional capacity. So these are two measures or two metrics that you have to judge if the drug is working or not. Now, there is this like caveat here that if you have one standard effect on cognition alone, that may be sufficient. So these are like guidelines in order to those who are doing drug development to have a target product profile. So if you will be able to have a product which will achieve either on both of them you have 0.5 standard deviation difference from placebo, or just on cognition, that is supposed to be middle effect, and this one is an overwhelming effect. And then maybe even if you are not, we are not able to show that you have a functional capacity, what is required, that should also or can be also considered as a confirmation type of package. So cognition measure, there are different attempts, and this matrix group is developing this endpoint. So it's not about biomarkers, it's now about developing endpoints for measuring treatment effect. And they came up with these two core primary endpoints. The first one is called cognition measure. It's like they are a battery of composite endpoint. It's a score. It's not latent variable dichotomized. It's a much more complex multidimensional measurements on the patient, and then it's coming up with a score for that one. And functional capacity is another type of impact of the drug on the patient behavior, which is measuring the functional capacity of this patient. So when I was at Wyatt, we were asked to help the clinical team to develop a drug which was coming from the early development with the potential maybe that that drug might be good in this type of indication, although the drug was also considered for other indications. So all the issues of early development of this drug are not of interest at this point. So safety, toxicity is already kind of accumulated for this drug. Now the company is entertaining the idea of marketing this drug in this indication. And the original strategy was the following. They will do phase 2A type of study, 2 doses perhaps of this drug and standard of care with a very short endpoint, 8 weeks. And then looking maybe which one of these doses will be the one which shows some promise, is safe, tolerated, and then they will continue with now a 18-month study duration. Each patient will be treated for 6 months. And then you remember that the requirements from FDA is indeed for submission you have to have this 6-month endpoint. So in a sense this study was a mini phase 3 type of approach. You do now not a very powered study like you will do in phase 3 with thousands of patients, but how about we will do a small size, maybe with power 80%, not 90%, maybe type 1 error, not 5%, because FDA will not just be reviewing it, but maybe 10%. So that was the idea of the company. And they measured that the overall timelines will be like this. So this is like beginning of the study, and it will take them, the start of phase 3 will be somewhere here. So in discussion with the clinical team, finally we came up with the following. Maybe we can combine these two studies in a single study and try to do the following. Investigate not only 2 doses, which they had initially, but more doses. maybe give more doses opportunity to be tested in this study. We put a kind of a compromise between 8 weeks and 6 months, working just with a 12-week endpoint. And one of the medical researchers in the clinical team is a member of the Matrix. So he was our link with consortium development. And at that time when we were discussing, he brought back from their findings that a 12-week endpoint might be good for screening drugs. So if a drug doesn't achieve any effect by 12 weeks, there is a very, very little chance that this drug will finally be effective at 6 months. So that information was taken into consideration in designing the study. And you can see here that the proposal is considerable one. You cut this type of about 22 months in development, which is very, very important if you think about cost-effective drug development. So the original strategy was better than this one because it was supposing to replicate in that second study almost the same situation as a phase 3. So definitely if you find that that drug will be working in that second phase study, you have much more confidence that it will be successful in the phase 3. But the pro of the revised strategy is that you can do that much faster. And the cost savings, of course, is in about millions. Of course, we are here probably not to count money, but for a company, believe me, that's a very important metric. Okay, so key elements of the study. So goal to do a single phase 2 study, and in order to accumulate enough information as a package for submission at the end of phase 2 study in discussion with FDA, we decided that we need to definitely not to consider just 2 doses. That will not be sufficient. And you will see that we consider 6 study drugs in this study, in this trial, and placebo. Placebo here is like standard of care. I will use the word placebo, but remember that there is no placebo. It's like standard of care, and then our drug is on top of the standard of care. So treatment duration, like I said, will be 12 weeks, and that was in collaboration with the Matrix group, sufficient predictive value for decision in order to go to proceed to phase 3. Co-primary endpoints, like I said, that will be changed from baseline in each of these 2 endpoints. Now, what is the conventional design? Conventional design will be maybe 2 or 3 active doses and placebo. So we considered as a benchmark, let's consider that we will do 3 doses of that one. And so we calculated what will be the requirements for that type of study. And it will be about 70 subjects per arm, will achieve good power in order to this will be the requirements or risk control. So you can see here already that this is not about p-value for rejecting the null hypothesis, where the null hypothesis means that the mean difference between endpoints was standard of care and our is 0. So probability of go decision is controlled not at difference of 0, but at 0.2. So that is like the team was very cautious of moving forward with development of this drug. If we just simply reject by a p-value smaller than 0.5, the null hypothesis, the drug is not the same as placebo. So you need to have at least, even for treatment effects of 0.2, you would like to go forward not more than 20%. So that's one difference with the traditional setup of hypothesis testing, if you like, in this setup. And of course, if it's 0.5, and you remember the requirements for phase 3 is, if indeed the drug is achieving 0.5 on each endpoint, that's a good drug. And so you quantify that, saying that probability of a go decision should be at least 80%. So I just want to mention here that there is even for traditional design, it's slightly different than let's say, conventional way of doing conventional design. Now, adaptive design, on the other hand, because we have 6 active arms, and we would like to have the same total sample size as the benchmark, We entertain to do 6 doses, but in order to be able to do it and do it feasibly, you have to do some adaptation. Otherwise, if you just do parallel group design for 7 arms, it will not be helpful. So we will use then response adaptive allocation across these 6 treatment arms during the trial. And I will have more details about how that will be implemented. We will have also stopping rules in case we early on, before we finish enrollment, we see that none of these doses will be promising in showing superiority with standard of care. We would like to stop the whole trial. And although we are not planning to stop early for success, because that, remember, that will be the only package which the company will go to FDA as at the end of phase 2 meeting, and usually you have to have substantial evidence in that package that drug works. So stopping early for success is not going to be a viable option, so we removed it, although in principle you can put a stopping rule for efficacy. But for the operational efficiency of this design, that the clinical team was asking to provide them an information about to start preparation for phase 3 or not waiting until the study will be over and then start preparation for phase 3. That preparation for phase 3 requires some time and some effort. So they were looking for statisticians to give them a hint or a trigger of start of phase 3 activity. So they will not start at that interim analysis, but they will start preparation for that. What that means is not the topic of today's discussion, but I will show in my simulation how good was the design in triggering when is needed to be triggering the start of phase 3, and how many times wrongly was triggering. It may be that finally this trial will be a futile trial, but you triggered activity of starting a phase 3, but that is something not so much importance. So now, how we can do this response adaptive allocation? Of course, we have to rely on some modeling, because just doing parallel pairwise comparison is not going to help you to achieve the objectives. So we will use a so-called normal dynamic linear model with non-informative priors in order to try to enhance our decision making at the interim analysis. And I will have some slides, but we have here a lot of people which are experts in normal dynamic linear model. And because the endpoint is week 12, but at each interim analysis, when we will look at the data, we may have, not everybody will have 12 weeks, but we'll have a good proportion of subjects which will have week 1, week 4, and week 8. And indeed, the measurements we're taking on subject level at this time point. So we would like, through a longitudinal model, to use that information in order to increase the decision-making capability at the interim analysis. And also, it's important in this type of indication, these are schizophrenic, almost schizophrenic patients, and of course the dropout rate is considerable one. So we were given this 25% expected dropout during the period of patients being in the trial. So normal dynamic linear model, why we use this one, the clinical team was not able to tell us if they believe in monotonicity of those response on each of the endpoints. So if, of course, if you have that assurance, you can use, let's say, 3-parameter logistic or 4-parameter sigmoid max. In this case, they were not willing to take the responsibility that indeed the dose response will be monotonic increasing. So we rely on this normal dynamic linear model, which doesn't put any restrictions on the shape of those response. It may be monotonic, may be not monotonic, does not developed for analysis and forecasting of time series data. There is a nice big large book by West and Harrison, and this methodology was first used for adaptive dose ranging study in the famous Aspen trial from Pfizer, and we have here I think in this conference 4 authors of that paper. So what is NDLM model? Briefly, observation on a patient assigned to dose D has a mean theta sub D plus some error term, and this error term is normal with mean 0 and variance sigma square. Now this mean at dose D is itself sampled from a population of means with mean mu 0d and variance nu square 0d. And then after that, let's say this is the mean at the standard of care, which is like you can consider it, this is your drug with dose 0. But then for the next dose, which we consider, the mean is nothing but a linear shift from the previous one. So you see, you sampled from a population of means which has the mean of the previous dose plus another variance. And that variance is proportional to this constant tau square, which is governing the variability of moving from one dose to another, but also is direct proportional to the distance between doses. So Vd plus 1 and Vd, this is the physical amount of drug at dose index d plus 1 and dose index d. So then in order to embed this in a Bayesian type of analytics, we need some priors for this one, and these are the priors. All variance components are inverse gamma, and all kind of mean components are assumed to be normal. And for example, here is a parameterization of inverse gamma, tau n divided by 2. If you don't want to be Bayesian, a way of looking to it is that you will sample, let's say, sigma square, or you will learn about the sigma square from a previous experiment, which was done with sigma n subjects. And in that experiment, you estimated that the mean of that variance was sigma mu divided by 2. And that's a very, very easy way to interpret and talk with clinicians in order to explain them what that means. And also, even if you indeed go and look at the data from a previous experiment, you can downweight now the contribution of that prior in your current trial by downweighting the sigma n. So if it was 100 subjects, you can say I downweighted by 10, and then you'll use 10 here. So that is how you downweight the information from the previous experiment in order to control the informativeness of your assumptions. Okay? So longitudinal model, how we are using, very simple. We are doing, as Einstein was saying, that do it simpler as much as possible, but not simpler. So we will use a linear type of piecewise linear longitudinal model. So y sub t is the observation of the subject at the last endpoint. In my case, was 12 weeks. But if I have on that subject's observation at week 8, for example, this is wt. So the distribution, conditional distribution of that observation is nothing but normal with some intercept and some slope multiplied by that object. And you do this for each time small t. Okay? And then these coefficients are themselves sampled from a population of coefficients. And as usual, everything which is on the mean scale is with a prior of normal. Everything which is on the variance component is a prior of inverse gamma. So you can do more sophisticated models, longitudinal models, and you can use more sophisticated model in your simulation exercise in order to see how this simplified piecewise linear model in the design is performing. And for example, one here you can think about, and I'm now using the terminology which was used last week when we had mixed effect model. You can think about that for a subject which was on dose D, subject I on dose D at time t, the observation is nothing but a mean for that dose, a general mean, plus a subject effect, and plus noise, but then is discounted by this coefficient. And this coefficient depends on t and the maximum t. Maximum t in my case is 12 weeks, and t might be one of the others, 24 weeks, sorry. So this is like if I keep constant this k sub d, in this case is minus 0.2, but I change the doses, that is the trajectory, mean trajectory of this mean of in time. This is time, and this is for given theta, what is the mean of that? Not this theta, but the whole mean of this object. On the other hand, if I keep theta fixed, but I change the k, you can get this type of shapes of time behavior. So we can use this type of real, if you like, or act that this is what will be in reality, but then apply the design which assumes that your longitudinal model will be piecewise linear, and then through the simulation you can see how good that design is. Now back to my problem. That's like kind of almost introduction to this type of adaptive design. But how we deal now with those two endpoints, and remember that the restrictions, regulatory restrictions on those endpoints are not similar. So on one you have to achieve at least 0.5, but on another one, if you achieve on another one just 1, that's sufficient. So we are using a utility following advice which we got here for many previous talks. So if this is a cognition endpoint, and f is the functional endpoint. So if you create this type of utility, this will be isolines of that utility function. And you can see here that if I am 0.5 on cognition and 0 on this one is 0.5. But in order to be 0.5 with cognition 0, I have to be at 1 with the function. 0.3, which was used in in the traditional design, is sitting just above here. So that is something which is you would like to be able to say, if it will be 0.3, I definitely want that this drug to go forward. If it will be over here, definitely I need to guarantee the design is very good. But if this one will be somewhere with the 0.2 and 0.2, and that's much smaller than 0.5 in this region, then I would like to make a go decisions no more than, let's say, 20%. So this is now how we, at each interim analysis, we will observe both endpoints, but in order to guide ourselves in to change allocation, stopping the rule or whatever, we will use this utility in order to guide us in decision making. So we will consider maximum 300 subjects. In the traditional, you remember it was 280, but 280 is without dropout. We consider here in the simulation 300, but we will implement also dropouts. So you expect that you will end up not with 300, but somewhere in the region of 270, 280. So they are kind of calibrated on the maximum number of subjects in the trial, and that let's see how one performs against other. So we have two targets for the design. One is find the doors which achieve the maximum utility. So that is denoted in my other slides, MUD. But also we would like to be interested in seeing, because that's a requirement which usually regulators will ask you when you go to the end of phase 2 meeting, to see how much evidence do you have that you know what is minimum effective dose. So another target will be this minimum effective dose, and if you translate that in the language of utility, that's the dose which has, or doses in our case, might be, that has utility 0.5. So the design itself, you will see here that adaptive randomization doesn't use this target, but in the simulation we will quantify how good the design was in identifying the MED. So allocation rule will be the following. So initially we start with equal allocation to all these doses, 10 subjects per dose. And then after that, allocation to placebo is always kept 20%, but it may be going higher if indeed we will observe that the variability on the placebo is higher than we expected. So you allow the placebo to have at some interim analysis more percentage, but not more than the dose which is maximum utility, because otherwise you can use all the patients on placebo, learning on placebo, and nothing on your drug. So that's kind of a flexible way how you impose conditions on the placebo. And then allocation ratio at each interim analysis to a dose D. This is, if you like, in the terminology of design of experiment, this is the weight of the design point D. In this case, is a dose. And it's proportional to the variability, direct proportional to the variability of the mean at that dose, normalized or inverse proportional to the number of subjects already at that dose, but then multiply by this probability that posterior probability that that dose is indeed MUD. So what the allocation is trying to do is to shift the allocation to the doses which are believed to be MUD, but also depending on how many subjects you have there and what is the variability of the treatment effect at that dose. So it's not that each subject will be allocated to one. So at each stage, imagine that you generate this vector of weights, and then the next patients will be randomized to all the doses proportional to these probabilities. So this is not an intention to treat design. It's not every time you put a subject on a dose. It's just every time you update the design and you allocate the subjects for next week, for example. In our case, it was 2 weeks endpoint, 2 weeks interim analysis. Every 2 weeks, you will replace the randomization list with a new randomization list, which is updated according to this formula. Okay, so parameters of adaptive design. We will have no stopping for success, like I said. Trigger phase 3 preparation when the probability that utility at maximum utility at that stage is greater than 0.5 is 90%. So overwhelming evidence in the data that you found, or you are sitting now on a dose which you believe that is a maximum utility function, and that posterior probability is greater than 90%. Stopping rule for futility, on the other hand, if that is very small, it's only 7.5, you will stop for futility, but not earlier than first 100 subjects. You don't want to react to the high variability in the data early on in the trial. And then go, no-go decision at the final analysis will be the following. If you will find a dose at estimated MUD dose, if probability that utility is greater than 0.5, which is that borderline of good drug, is greater than 70%, you will say go to start now phase 3 and go with this trial in a large number of subjects. It will be a failure if that condition will be satisfied, and then of course you can end up very easily with some kind of inconclusive. And instead of saying that it's black or white, we categorize in success, go decisions, no-go decisions, and then this one is inconclusive. And it's now up to the clinical team having access to all the data to make the decision what they want to do. So we will not quantify in simulation the performance, but we will quantify how many times the design will achieve the so-called inconclusive decisions. I have to control my time.
You have to.
Minus 2 minutes. Okay. Sorry about that. So then how we do the simulation? So you run the simulation on a bunch of what-if scenarios. And so in our case, we had about 12 scenarios. The slides will be available. Who wants? I can give them. And then you look at the performance of these designs under different scenarios. So I have in my set of slides, I think about 6. But I will go then directly to this final slide, and I will finish. So this is now not graphical. The plots are showing you how the design was allocating subjects and how often you found MUED. But here is, for example, this is a situation, a scenario when this drug is just like standard of care. And as I mentioned, you can see that stopping for success in this situation will be just 0.08%. So it's not type 1 error 0.05, it's 0.0008. But this one is the one which is in our situation was like a null case. And you can see that in that case, you will, with this design, you will just move forward or make a wrong decision only 6%. The restriction was 10% here. The other one, you see that it has 9%. And then a scenario which is good one, this one, you can see that you achieve a success at the end of the study in about 83%, and you are doing it with 293. This is the sample size in one and another one. For a scenario which, for example, this one is good on cognition, not good on function, the design is very powerful, while the traditional, of course, is losing because traditional was targeted 0.3, 0.3, didn't take into consideration this asymmetry in the trial. This one is a scenario where you have an increase and then decrease. And we wanted to see how this NDLM will pick up this type of tough scenarios. And you can see here that it was doing a good job. It detecting it in about 91%. So conclusion, team needed to trade off this additional burden of running a flexible design versus answering research questions more efficiently. And I think that through the simulation, we were able to convince them that this design provides them additional benefit and some additional efficiency in their program. The advantage of using adaptive design in this CNS Learn study where that we cannot use this monotonic dose response, and that parametric or model-based designs with simple 2-parameter, 3-parameter will not be possible to be used, and so this is why we relied on this NDLM model. So I will stop here, and I will take one question, and then and then we will move to the next session, next presentation. Yes.
In a prior, you had a variance which was proportional to the difference in doses. I think you said. So are the doses increasing?
Doses as a physical amount, yes. They are ordered in an increasing amount of dose, yes. So let's say 10 milligram, 15 milligram, 30, and so on.
Quick question. What happened to the trial?
The trial, this was a wired clinical team, and when we were almost ready to run, Pfizer came on us.
Okay.
So I don't know what is going on with this trial now, but because I'm not working anymore in wired.
It's a sandwich and closed it down.
Okay, so next talk is by Luke Ronzato. So again, sorry that mine was not so academic, but I guarantee you that Luke will be very academic presentation.